Well, hello everyone. Good afternoon, good morning, or good evening. Where are you? Anywhere you are. My name is Yasi Ghadar. I am going to be lead application for this year's student cluster competition at the SCC21. Today we have Robert Blake with us. Robert Blake received his bachelor in computer science and math from Tulane University in 2004 and his master of computer science and numerical analysis from the University of Illinois Urbana-Champaign in 2010. He's specialized in numerical analysis and parallel algorithms. He has over 18 years of experience in software development and 16 years of experience in scientific computation and 13 years of experience in cardiac modeling. He was the co-founder of the CardioSolve LLC, a company devoted to bringing basic science into clinical application through scientific computing. There, he implemented a system to guide cardiac ablation procedures based on HPC simulations of patients' anatomy. His major accomplishment include the development of 4 parallel cardiac simulations, the development of computer language for modeling membrane kinetics, and he has also worked as an independent consultant for Medtronic. So today he's going to give a talk about the cardiac and how you use it. If you have questions, we suggest you to post it in the chat, and I think he has implemented some break that he will go through the questions and answer them. Thank you, and thank you everyone. Robert, please take it away.
Hi, thank you so much for the introduction. I'm just going to give a very simple introduction to what cardioid is and why we do cardiac model in the first place. Then we're going to move on to how the cardiac simulator works and things that you will need to be aware of when you're running it as part of your student competition. So just very, very briefly, why do we do cardiac modeling at all? This is a good comic here. It's really funny. You know, here's an example of what you should never do with your heart. Cut it in half and look inside. The truth, there is some truth to it though. Like the heart is very, very difficult to observe. If you want to do anything to it, interventions can often be life-threatening. And the whole idea of using simulations for cardiac science is that maybe you can get simulations that allow you to cut inside and look inside the heart, which you can't really do clinically. And this can be used for everything from doing basic science to like making better implanted cardiac defibrillators to planning which drugs are going to affect the heart better. You can plan surgeries. You can sort patients on whether they are high risk for a cardiac event to low risk for a cardiac event. You can basically, in a simulation, you can torture a heart all you want and not have to worry about harming the patient. So that is why we are primarily interested in doing cardiac modeling. Now, the thing you need to understand about the heart is it is both an electrical and a mechanical organ. Every cell of a heart is as electrically active as a nerve cell. The only difference between cardiac cells and nerve cells is that cardiac cells aren't myelinated from each other, so they don't have like the wires of insulation. But the way that it works is when a cardiac cell fires, it flashes and it sets off all the cells near it. Here you can see kind of a graphical representation in 2D of what this looks like. Here we're activating all of the cells in one row right here electrically, and that causes like a domino cascade of waves traveling through the heart. And these sorts of patterns inside your heart correspond to, say, normal sinus rhythm. However, in various strange situations, you can end up with something called an arrhythmia. This is what it looks like on EKG. This is literally what it looked like if you took a voltmeter to different sides of your chest and just touched it. You would get these measurements in the millivolts for normal rhythm and these measurements in the millivolts for a ventricular tachycardia. It's caused by electrical patterns that happen like this inside the heart tissue. Here, things have gotten wedged in such a way that the activating pattern sets up a never-ending loop that always goes on, and this corresponds to having a heartbeat of roughly anywhere from 200 to 250 beats a minute. Your heart kind of feels like it's fluttering, and if you leave that sitting long enough, it can devolve in just complete chaotic behavior, where these spirals are happening all over the place and chaotically. And like we can see this when we observe externally on the patient with a voltmeter. Just you have like very, very, very random looking ECGs when you come out of this. So these are these three main modes the heart can work in. This one you will survive, but you will faint. But if you get this pattern happening inside your heart has effectively stopped beating, and you will start to experience brain death in 6 minutes, and you will be dead in 12. So these graphs happening right here aren't from simulations. These are from actual clinical recordings called optical mapping recordings. The way they do this is they take a piece of tissue, they inject it with a voltage sensitive dye, and then they shine fluorescent light through it to get a movie of what it actually looks like when a heart activates. So here you see, this is a real recording of what ventricular fibrillation will look like, or what ventricular tachycardia looks like. This one over here is a real piece of tissue with what fibrillation would look like, just random chaotic patterns happening normally. Now, the challenge with all of this, in order to get these pretty graphs, you see mostly what the only thing clinicians ever see are these line graphs you see up here. In simulation, we can see everything at all points in time, but even to get these pretty pictures right here, you have to take if you want to do this to a whole heart, you have to take the heart, you have to suspend it in cut the heart out of the animal, suspend it in a fluid that has this dye in it, shine lights on it, and then, everywhere you are, put cameras around it, and hope that the heart doesn't die within the next 20 minutes while you're doing your experiments. Whereas with the simulation, we can just look at everything at all times. So this is a simulation of what normal sinus rhythm looks like with the electrical and the mechanical coupled together. As you can see, the electrical waves start from the inside, they proceed to the outside, and the whole heart contracts as one whole motion, trying to push all the blood out of the ventricles. This is what it's supposed to look like. But you can end up with simulations that look like this. Here you're going to see a flash of red, which corresponds to a premature shock we've delivered. And then you end up with ventricular fibrillation like this. If you look very closely in this picture, you can see the heart trying to contract, but because nothing is synchronized, the heart is kind of locked in place. And this leads to the sudden cardiac death symptoms that you can see in actual patients. So just as a side note, when this behavior happens, the way clinicians fix it is they deliver a huge shock to the torso of the patient. And the whole idea is to degauss this behavior, return it to a normal pattern where the wave can finish, and then normal beats can proceed like normal. Are there any questions on this so far? I also have to say all the images look really, really cool. The modeling and the visualization of all this stuff looks really awesome. Okay, so the heart, it requires multi-scale modeling in order to work. We're interested in simulating hearts, which can range anywhere from like 10 centimeters down to having to simulate at 100 micrometers. Heart is roughly about the size of your fist, sitting in the center of your chest. The time scales that we want to simulate, we want to simulate anywhere from, say, 10 heartbeats, but our time steps have to go all the way down to, say, 10 microseconds in order to get accurate simulations. In order to get accurate simulations of these things, you need somewhere as many as 400 million cells to get a fully resolved behavior. And this can lead to 8 billion degrees of freedom when you're simulating these things. And the way these simulations work is we have a heart that we usually get the mesh for from an MRI. The continuum in the heart is modeled with a monodomain bidomain discretization. And that at every single point in the heart, we have a complicated ordinary differential equation that for each cell monitors how ions flow through the cell membrane, and different ions flow through different kind of these ionic channels. We'll get into that in a second. So we'll first talk about the continuum math here. The bidomain model is just this representation of equations right here. It ends up looking like a reaction to fusion equation. Here we have this complex tissue. This is a close-up histology of what heart tissue looks like. And we represent it with this kind of electrical schematic for how we simulate this. So we have a whole bunch of a field of resistors on the outside that corresponds to this external conductivity. And inside, between the cells, there are these little subway tunnels that connect to the internals of the cells called gap junctions that allow ions through. And we have a whole bunch of internal resistances this way. You should also know that electrical current prefers to flow along the heart fiber and doesn't like to flow as much against the heart fiber. So in this instance, we would say tissue would want to flow this way because the resistance is smaller, and would not want to flow this way as much because the resistance is much larger. The difference between these two things is called the transmembrane potential. And you should know that heart tissue, just like nerve cells, usually sits at a negative transmembrane potential. That is that the internal is at a different voltage than the external of the cell. And when things pump from the inside to the outside of the cell, it sets up a whole cascade reaction that causes the pictures you were seeing before in the previous graphs. But this is the rough schematic you can think of. We have a field here, it's anisotropic, and we have these little current pumps that take current from one field to the other, or vice versa. Now, the way these pumps work is we have this ionic membrane there's a capacitance to the membrane that's just given by a simple RC circuit. And then there's this extremely nonlinear ion term here that has all of these differential equations. We roughly represent it with a schematic like this, where here's the internal potential, here's the external. We have a capacitance, and then we have a voltage source. This is what's called the Nernst potential for the cell. This is kind of what the ions want to do across this cell boundary. And we have this resistor that's governed by these extremely nonlinear terms here. And inside the cell, you have a whole bunch of ions trying to get internal to the cell, and you have these Stargate-looking contraptions embedded within the cell membrane that under various conditions open and close. And when they open, they're sized in such a way to only allow specific ions through. And so we represent all of this with a state vector, and the whole differential equations for the ODE portion of this computation is done right here. So just as like a fun little aside, I had a company where we built a system. If I could get an MRI of your heart within 24 hours, I could actually recreate your organ on a supercomputer and run clinical interventions and on that supercomputer and predict what the outcome would be. And we had a company where we would take those outcomes and deliver it to clinicians within 24 hours to show, like, if you do this to this patient, this is what will happen. And just to verify like how this works, I volunteered for an MRI study where I got an MRI scan of my own heart. Just as a quick note, like if you want to go and do this, if you ever have to go in for an MRI for either your brain or your heart, if you bring a USB key that's still in the package and ask them for the scan, most clinicians will give you the scan. They won't accept any USB key because they have to worry about malware getting onto their computers. But if it's still in the box, they'll most likely just transfer it to your USB key for you. So I got a scan of my heart. I ran it through some segmentation software to just show, this is what's happening for my heart. We ran through some software that we had, some algorithms that I've written to, from those scans, reconstruct the fiber orientation for my heart. You can see it makes this cool twisty motion as you do, and this is really important to get the correct electrical and mechanical behavior. And then I could go and run simulations of this is what an apical beat would look like in my heart. Here you see I put a stimulus here and see how things would progress. And go ahead. Sorry, I thought I heard a comment. So this was really, really cool. We had a system set up where once we got an MRI, we could start doing simulations in under 2 hours. And we had a 24-hour deadline because the patients would get the MRI the day before and then go in for a surgical procedure the next day. There's lots of fun stuff you can do with cardiac modeling. For example, you can use a heart model embedded within a larger torso, and then you can put electrodes on the torso to replicate what the clinician would see if they were doing an ECG. And this is various ways of doing that represented here. But in this way, you can actually say, hey, if you do these simulations, this is what you would expect to see when you're doing an ECG looking at this particular patient for their particular cardiac geometry, because everyone's heart is different. Likewise, for drug simulations, you can do a lot of fun simulations. Here is a simulation where we took a normal heart with an extremely high resolution heart taken from a 14-year-old boy called Lewis as part of the virtual family data set. And we took this high detailed resolution of his heart. And in one heart, we left it just normal heart contraction and electrical response. And in the other heart, we took one of the channels and completely blocked its behavior. And this channel is extremely important for the heart being able to reset itself properly. And because the heart can't reset itself properly, what you can see is that you end up with these waves that go around in a circle. This, what you're seeing here is the isochrone of where 0 voltage is. And so what you're seeing is a wave that's going kind of around in a circle from -100 to 40. And it's repeating over and over again. This patient's experiencing what we call torsade de points. And you can see on the corresponding ECG for this patient, this just looks like kind of a twisting of a ladder if you do it for long enough. This is what's known as IKR block. And it's why all drugs have to be tested for cardiac side effects, because if a drug even touches the IKR channel, 10% of the population might die from taking it due to effects like this. And you can show how this happens in simulation and look in far more detail in simulation than you can ever see in an actual patient. Because in order to observe this in a patient, you have to stick plunge electrodes through the heart, and that kind of stops it from beating, which defeats the whole purpose. So that was kind of the rough idea of the science behind what we're doing. Before I go on to the technical details of the simulator, are there any questions on the science? Okay, I'm going to go launch straight into the technical details then. Feel free to write questions. I can talk your ear off about this stuff all day. It's really cool. So Cardioid was a Gordon Bell finalist for supercomputing. If every heart has roughly 400 million elements and 8 billion degrees of simulations, 1 second of simulation typically requires anywhere from 50 to 100K time steps, and each time step requires about 2 billion math function evaluations. Despite all these constraints, we got Cardioid, before I came to the lab, they got Cardioid to scale to all of the supercomputer Sequoia in almost real time. So being able to do all those computations in real time, which is a massive, incredible feat. Before I came to the lab, Lawrence Livermore National Laboratory, it would take me a month to simulate like 5 heartbeats in full. Whereas here with a whole supercomputer, they can do it in real time. However, this is extremely strong scaled. And if you actually go and look through the code, you will see echoes of this. At its limit in the strong scaling, they only had 200 cells per thread. And they got rid of all the math functions like exponential and logarithm, of which there are a bunch, and replaced things with rational polynomials. There were hard-coded vector intrinsics that were specific to blue gene Q in every critical loop. Because they were so strong scaled, MPI was too slow. Instead, they used DMAs on the network interface and used thread barriers based on L2 cache access for that individual machine. And when I first got to the lab 4 years ago, my job was to convert all of this to work to GPUs without losing speed. So things you need to know, we did get that to work. Things you need to know about the performance characteristics. Cardioid uses a finite volume discretization on a regular grid for its simulations. However, the grid, while the grid is regular, the nodes that are kind of active on the grid depend on the geometry. For your simulation, you're going to be using square or rectangular slabs of tissue, so you don't have to worry about that as much. But for actual cardiac simulations, we have to do quite a bit of work to take a patient MRI and reduce it down to a regular grid and note which voxels belong to the bath, which voxels belong to the heart. There are 2 main modes that are used during computation. You have some spatial diffusion that takes about 50% of the time step. This is a 19-point stencil We have different implementations of it that are optimized for CPUs and GPUs, and you can switch those out at runtime. This is the part of the code that requires MPI communication. The parallelism seems to be limited by network latency. I know for a fact that when you run this on Ethernet in parallel, it will only scan to say like 4 or 5 processors. You need an InfiniBand switch in order to really take advantage of the small messages that are being sent back and forth. The GPU performance of this portion of the code is limited by the memory bandwidth. The other half of the code, where other 50% of your time step goes, is in solving the reaction ODEs. That's that membrane complicated ionic term. This part is computational bound on both the CPU and the GPU, and it is embarrassingly parallel. So it parallelizes really, really well, which is one of the reasons they wanted to use it for the Gordon Bell, because it's rare to find a problem that has so much computation that you're computation bound. And when you're running this, depending on how you want to run this on your cluster, there are different drivers for each of these two modes that you can, for example, use CPU diffusion and GPU reaction ODEs. You can switch these out and use whatever you would like to get the fastest speed for your code. The Stencil computation, it's network communication interleaved with computation. It's a whole bunch of MPI iSends. It's a 19-point Stencil, and it's heavily optimized. We have some naive implementations, but we also have CPU versions that use OpenMP and things that use vector intrinsics for faster computation. On the GPU side, we have shared memory blocks with polyhedral loop optimizations for extremely fast computation of large blocks of code for this. The fastest version currently uses Volta-specific instructions. If you aren't using the Volta-specific instructions, there are slower versions that still give you good speed, but the absolute fastest is pretty much hand-tuned for Sierra. For the reaction ODE models, you need to know that this portion of the code is embarrassingly parallel and compute bound. In general, in real simulation work, the reaction ODEs are different for every single experiment you want, and they have to be optimized by hand. None of you are going to be doing this. This has all been done for you. Just know that when you're going through the code is going to look extremely nasty, and don't worry about that because the code is auto-generated. I wrote a computer language to take a high-level description of the differential equation. For example, here is the gate for an IKS channel, and you can see it has these horrible expressions here with 1 plus x of 5 minus things with magic numbers embedded all over in them. And from these equations, I can go and use some generated code with SymPy to automatically differentiate these things, use specialized numerical methods to integrate these things. And in particular, when you're going and working on NVRT, when you're working on CUDA calls. So maybe back up, when you're working in CPU mode, I have code that generates rational polynomials and will generate SSE instructions hand-coded by hand to solve these things as quickly as possible on the CPU. When you're on the GPU, I have code that will go and use NVIDIA runtime compilation to embed some of these coefficients directly into the actual object code that's run on the GPU. And the resulting code looks extremely nasty. When we drop into a terminal, I'll show you what some of it looks like. But we did that because that gives you a 10x speed up. But that might cause problems if you try to move to AMD, and we might have to talk together to make sure I can give you a version of this that runs well on AMD systems. So things that we have to do, depending on what system we're on, we have to do different things. Replace things with rational polynomials. On, say, a standard CPU system, we have to do this. On a GPU, we have to do this. If we're using Knight's Landing, you don't. We have to use fission or fusion based on how many threads we're using and what the system supports. Sometimes the system Xblog is way too slow. On BlueGene, that was the case, so we had to replace it. We have vectoration with Intrinsics that has to be done on any CPU-based system where it isn't done on GPUs. And the layout of all the data structures has to be changed, whether you're running on CPU or GPU and the exact system you're using. And there's code in there to switch the layout of internal data structures according to that. So just to let you know, when you look inside the code, you're going to see a whole bunch of horrible magic numbers here. And go back. let's say I want to compute this expression right here. It makes a nice like little step graph right here, and that's what I want to represent. Well, there's no reason I have to use 2 divisions and an exponential to compute that. What I can do instead is just compute a rational polynomial that approximates that, and then use some very simple polynomial loops with like only 1 division to represent the final result. And that ends up being much, much faster than invoking exponential or log. And we can do that for a lot of the functions in the code. Likewise, in the GPU, the reason we had to resort to runtime coefficients is that if we're trying to evaluate, in many cases, we don't know what the coefficients will be until runtime, because the coefficients depend on the time step you're using. And if we put the coefficients into an array, the code for, say, like the simple loop just computing a polynomial will take about 200 microseconds to run. Whereas if we embed the polynomial coefficients directly into the code, it runs about 5 times faster. And this just magnifies the more that you do. And in fact, you can look to see this is just roughly the same cost as a mem copy. So, and when you actually go and look at the assembly, you don't get for loops. You actually get just raw FMA instructions with coefficients being read out of constant memory. So that's why we're doing those horrible things to the code inside the run things. We also do some tricks on the CPUs to, we just use simple duff devices to evaluate polynomials to avoid the number of loops we have to execute. This ends up giving some speed benefits here for very little cost. And we have hard-coded intrinsics for various platforms, which we need for some systems and not others. This is the structure of arrays thing we convert, whereas either we do structure of arrays where we say like, here's all of state variable 1, here's all of state variable 2, and n is the number of cells we have in the system. Whereas if you do arrays of structures of variables, you store a vector width for a single, say like vector width is 4 for most Intel systems. And then your state is this n divided by width, and you do operations on one width of vectors at a time. So on CPUs, this one is much faster. On GPUs, this is much faster, and the code has to be changed to correspond to that. So what we're going to be running here for the actual benchmark, I'm going to be running you guys through the Niederer benchmark. It is a specified system where you take a slab of tissue that's 3 millimeters by 7 millimeters by 20 millimeters. The fiber orientation is defined and the stimulus is defined, and you stimulate it and see how the wave propagates. And you should see curves that run kind of like this. And depending on the discretization of your mesh, you should see different curves. In addition, this is a nice problem because both the discretization can be tuned down, and you can just increase the volume of this box to get any size system you want to really execute and test out your clusters, see how well it's performing. And so my goal for here, I'm going to have you guys run a convergence study because real medical simulations depend on the waves propagating through the tissue at the correct speed. If those waves are off, then simulation results quickly are not useful for clinicians. And I want to see what resolution is required to get accurate conduction velocity simulations. So this is going to require you to have to run both a whole bunch of small simulations and larger simulations to verify what the conduction velocity should be for your problem. Are there any questions so far?
So far in the chat.
Oh, can you talk more about the challenges of AMD? So the main challenge with AMD is going to be that all the coding right now is written for CUDA. It's not HIP compatible. I can work with you to fix that and change that. So there's CUDA specific calls. That's challenge 1. Challenge 2 is there are some Volta specific instructions embedded in. There's some like NVIDIA specific things that AMD doesn't have. So for example, in the diffusion stencil, there are some atomics that only exist on Volta that we are using that I don't believe that AMD has equivalents for. And so you're going to have to use like a slightly different algorithm that's a little bit slower on AMD. The other thing you need to know is that the ODE portion of this is highly dependent on the runtime compilation. I can change this with your help and for you, but this will make more sense when we go and look inside the code, why you're going to have trouble getting this to run on an AMD system. So let's actually do that now, because that's the main bulk of the talk. The rest of the time, I was going to walk through demos of here's what the code, this is how to run it, this is how to build it, and where you might want to look if you are changing some of your code. Is everyone okay with that? Or are there more questions on the science before we launch into that part of the talk? So this will be a challenge for the cloud competition, which is on Azure. So this will run on, I forget, what system does Azure use? Is it AMD GPUs and Windows builds?
It's got AMD HPC, but it's got NVIDIA GPU. But if we have to do both a GPU CPU comparison, which was done for a couple of the applications last year, I guess if there's a parallel Intel system, we might need to work with you on what's possible on Azure.
Okay, so the
Yeah.
AMD, when I say AMD, I mean specifically AMD GPUs. The CPU stuff should work on AMD just fine. And I wasn't planning on asking for a CPU GPU comparison. I just want you to get the results the fastest way you can.
Okay, that's what I needed to hear. Thanks.
Yeah. So whatever mode you want to run to get the results is fine with me. The results are equivalent. I've tested it enough to know that results are equivalent between CPU and GPU. All right. Well, let's exit out of this real quick and open up this thing. All right. So compiling the code. The code exists already on SPAC. So you can either compile the code through SPAC or you can compile the code manually. The build system is CMake, and there are various build options you can use when compiling this. So if I wanted to compile the code manually, let me just run CMake here. I've already run it here on a system I have control of. Here I have said I'm just building the code. It's used from Lawrence Livermore, which is a library that helps make CMake easier to use. You do want to turn on enable MPI and enable OpenMP, and then build from there. The build system does require CMake and does require Perl. Inside, after you do that, after it's configured, you just type make, and it just runs. There are very few dependencies. It does depend on MPI, and it does depend on OpenMP and Perl, but that's about it. That's all you need to know in order to build it. So you should be able to see that here. I'm going to kill this right here so I don't have to watch it build. The other thing I want to show you here is what some of the code looks like. And this code looks horrible. I just want to forewarn you. I should also add, like, this code is on GitHub. It's open sourced. So feel free to look at it, modify it, do whatever you want with it. This is the code that has the core kernel that computes the reaction code. This is where 50% of your time is going to be spent. And if you actually go and look at this code, let me load it up in VI so we can get syntax highlighting. If you actually go and look at it, like, it's going to look pretty bad. So here's a whole bunch of code that's computing the rational polynomial and turbulence. This is all generated code. I would never maintain this by hand. that would be madness. And after we have all this code for generating the rational polynomial interpolants at startup, we then go to this code, which is horrible. So this is the entire C code for the main routine embedded inside a C string. So I see a question, MFEM is listed as an optional dependency, so it's not needed. No, there's actually two main electrical simulators. There's one that's MFEM based, and there's one that is finite volume based. For this test, we're only going to be, the MFEM one is currently under development. It's not really ready for primetime use. We're only going to be using the finite volume version, so you will not have to compile MFEM. There is, if you want to use CUDA, you have to turn on CUDA though. So you need to be aware of that. So this is the horrible code, and this is all of the code that we want to run embedded inside a giant C string. And you know, that's not great. So just so you know, like this code is also auto-generated. I wouldn't modify this code if I didn't have to. I use the melody thing to generate this. And this is the part right here, which you might have trouble getting to run on an AMD kernel, because here we are actually calling into the raw CUDA driver functionality that is not CUDA based, and I don't think there's an equivalent in the automatic HIP translation programs. So in order to get this portion of the CPU code running, or in order to get this GPU code to run on an AMD GPU, what you might have to do is instead of passing this to NVRTC, dump this to a file where you can print it out and just compile it and have HIP run it with the generated code and use that. But we can walk you through how to, if you talk to me, send me emails, I can walk up a how-to on how to do this and maybe even check something in that will make this work okay for AMD Citus codes. Likewise, if you're running in CPU mode, this is the version of the code that will run. It will use this SIMD library that I have to automatically load data from these places and run the code as if it's just normal C code. But all of these codes, all of these operations here, even though they look like they're working on doubles, they're all working on raw intrinsics for the system you're on. And it identifies what the intrinsic width is at compile time. So you don't really have to worry about it. And if you just want to, if it's not working, you can just tell to not use intrinsics and it will fall back to doubles. But you'll get like a 4x slowdown. So that is that portion of the code. Again, to build it, all I do is I run ccmake and I make sure that when I'm running it, I turn these things on. You also have the option of running it with SPAC if you want to build it. I find that easier in some instances. Here I have a env I have right here. And here's a list of the things I did to create this command. So I created the directory. I run SPAC env create.d. I activate the directory with SPAC. I add cardioid, and I'll document instructions to how to do this all on the main GitHub page, so you don't have to write this down. I tell it to find external packages. I tell it what compiler I want to use. And I tell it to, instead of going and downloading source code yourself, just get it from the directory above. And then I run SPAC install, and it will build the system for me. It does so by putting the system in the SPAC build directory. So I can go in here and type make all I want. And likewise, it'll put the output here in the SPAC build out.txt. So you can see everything here. Here are the configure args that it used. So you can see what args it passed into CMake. Here I'm running a version without CUDA. So CUDA is off. And it's running with OpenMP on. And it's running with enable MPI on. And you just do, let's cd to my environment. Spactivate this directory. SPAC install. and it will just go and build the program. It will also, if you don't have MPI installed on your system, this will also build an MPI for you. We do require BLAS LPAC, so this will build a BLAS and a LPAC for you, and we'll get all of that stuff working. So if you want to look at what the spec.yaml looks like, I've just used the spec external find. We'll find everything that already exists on the system, so it doesn't need to go and get it. It will then go and like, this is all the packages it found. It says I want to use the GCC 8 compiler because I need something that has Fortran and OpenMP. And then I'm using these compilers. And at the very end, I want to develop Cardioid from the directory above at master. And I type go and it will build it for me. MKL is fine for the linear algebra. You're free to use it. You should know, though, that it really doesn't matter. There's nothing inside. Cardioid requires it for setting some things up, but none of the hot loops use the linear algebra libraries. It's only for setup. There's not really any benefit to using any of the bigger BLASs. None of the hot loops are using BLAS at all. It's only, we just need something that can compute eigenvalues in the initial system setup. So anything works. It runs really fast, and after it's done, you don't need it anymore. And so, yeah, I'll fix the instruction in the GitHub. The spec directions are old. If you want to know more about using spec environments, please go and read the most recent spec tutorial. They have a whole section on using spec develop and spec environments that I also wrote. And I can walk you through that. It's actually a very, very simple process to set this up using spec. And it's really, really nice when you turn CUDA on, because I just do spec plus CUDA. I do Cardioid plus CUDA at master, and it will link it up to the CUDA that it found. It's really nice. So that's how to build it. For running it, I'm going to be making a directory here very, very shortly, and I'll send updates when it happens, but there are tests inside here for running Cardioid. Here we're going to look at, say, like the Niederer test, and we're going to look at the ray run. So the main input file for Cardioid is an object.data file. And this is what the input deck looks like. You can specify what kind of anatomy you want to use, what kind of parallel decomposition you want to use. There's tons of options here. What kind of algorithm you want to use for the diffusion, what kind of algorithms you want to use for the ODE, where you're applying stimuli, and various DT and time options. For the convergence study, I'm going to be generating a whole bunch of object.datas that change the DTs and change the discretization for you. So you don't have to define anatomies. This will actually go and define a brick for you. And the size of all these things are in millimeters. You can specify the size of the block to make it big enough. And this is how you're going to change kind of how big the problem is. And for diffusion, I'm saying here I want to run CUDA. I could also run GPU. There's open, red, black. There's a ton of different options available in the code. Some will run on different platforms. Likewise, you won't be messing with these. These are the conductivities. This is the reaction model we want to use. For most likely, you're going to be using better TTO6 because that's the one that is heavily, heavily optimized. Though if you make your own model to make sure that it runs well on an AMD GPU, you might want to rename this and make your own thing very, very quickly. Likewise, these define the stimuli within the block, and then there's some code here to print out the sensor. Now, you should know that when you build this, you will end up building two binaries. So let me go up here and show you the manual build. Inside bin, where is it? Oh, this never finished building. That's right. I killed it. Let's go to this directory above. So you'll end up having the cardioid binary that you'll run for building the thing, you're also going to end up with this single cell binary. This lets you run the single cell in isolation. And one of the things that does take a while when you are first running this is you need to go and, let me get this to do faster. One of the things that does take a while when you're doing this is it has to compute the rational polynomial coefficients for all of your operations. And that can take a little bit of time. You'll not want to do that during the competition. I'm going to be trying to give those coefficients for you, but those coefficients exist in this file right here. So inside this object.data file, you'll see a whole bunch of here. Here are the cached coefficients for what it needs to use for its various interpolation functions. And I will be trying to give those to you so you don't have to compute them, because computing those can take up to like 10 minutes when you're first running the program, and you won't want to do this for every single one of your runs. So these will be built into the object.data files that I'll be giving you guys. But once you run that, in order to run the code, I just do cardioid object.data and I'm done. It will automatically generate restart files if you run this from this thing, and it will just pick up the last restart file that was run and run it again. It's been heavily optimized so that if you need to restart a system, you can just rerun it and it will run to completion from where it left off. You don't have to change any arguments to the program. Everything's embedded inside this one object.data file. And when it runs, it actually dumps this file into the output so it can restart itself. It's very nice for running it when it's been accidentally shut down or run out of time on the cluster. Are there any questions so far?
There are a few in the chat.
I've been reading the chat.
Oh.
I haven't really seen any questions that I haven't answered.
Maybe you might have covered them.
Yeah, I don't know. I see like MKL question.
Yeah, the MKL question is the MKL, anything is okay for the linear algebra you want to use, but none of the linear algebra happens on the critical loop, so it doesn't really matter what you use. In fact, I'm not sure that the version you'll be using even touches any of the linear algebra on any point in the computation. At most, the linear algebra is used in the initial computation of a whole bunch of 3x3 matrices. So there's like nothing intense going on there. Actually, I think the interpolation does use some SVD algorithms, but you know, it's very easy. Like, and I'm gonna try and give you object.data files that remove all of that stuff as well. So. Short version is it won't leak unless you give it a LPAC, but it's not going to use it much, if at all. Any other questions? Any other comments or things you want to know about the weird world of cardiac modeling? The GitHub repo is at llnl/cardioid. So you should be able to find it there. The current SPAC setup instructions are out of date. I'll be updating them today to use the new SPAC develop workflow. I just haven't gotten around to changing that yet, but I demoed it to myself today. And so I'll take the instructions you saw on the command line just now and put them into the documentation. The other thing, if you actually want to get in further to it, there's another thing called llnl/melody. That is the tool that generates the source code for these models. You probably won't ever have to worry about it or run it. But that is what I'm using to generate the horrible better TT06 source code you saw. Oh, what size USB key? You know, the things are actually really small. An MRI, so it depends on whether you're getting an MRI or a CT. CTs are higher resolution than MRI. MRIs are that you get in the clinic are usually about 1 millimeter by 1 millimeter by 7 millimeters resolution voxels, whereas CT can go all the way down to say like 200 or 400 micrometer. So MRI scan will usually cost about 1 megabyte of storage. A CT scan will definitely fit under 100 megabytes. You don't really have to worry about that. It's pretty small, all things considered, like appreciable to the size of a podcast. So if you get a 1 gig USB key, you should be fine. They can do higher resolution MRIs, but they have to stop your heart in order to do the MRI scan, which is kind of a problem. They usually only do it to dead people, which is why we have such really good pictures of this Lewis character, because he was a kid who died and they just put him in an MRI scanner for like 5 days. Normally when they do an MRI scan of your heart, they put an ECG on you and try to scan your heart while it's still, because otherwise they can't get a good picture of it. Yeah, Osirix is really, really good. For segmentation, I typically use something called Seg3D for segmenting the tissue. I have people who have gone in to get their brain scanned, and I've helped them all go from like, here's the CT of your head to let's put your brain through a 3D printer so you can actual like print out of your brain. I also have sitting somewhere on my desk, right here. I can't show you because I don't have the video on because my webcam's broken, but I have a actual physical representation of my heart that I used to take to conferences, that I can literally like give you my heart to hold in your hand, because after we did the MRI scan, we 3D printed it off. Yeah, I know far too much about this stuff, and I can tell you like long stories at any point in the day. There's lots of interesting stuff in this field. So the results folder, I will write a description of how it works. One of the things you can do when you are looking at this, so let's go to here. It writes out time steps that can be hard to parse into something to visualize. We have some stuff for it, but it can be kind of annoying to visualize. The main thing you need to know, though, is that during a simulation, you can load different types of sensors. This is one of them. It's called an activation time. And this sensor in particular records the time where every single cell flips to its on state. It does that by recording when the cell crosses the 0 voltage line. And it ends up writing a single file that you can use to visualize when every single cell turned on. And from there, you can generate a nice, pretty picture in whatever system you're using. So let's run this real quick. I believe this one might be a little big, so let's modify it to make it smaller. Yeah, this guy's a little big. Let's make it a little smaller. Let's make it the base Niederer, and let's change this to 20 just to make it a little faster. And here, because I've changed reaction objects, it's going to have to go and recalculate all those coefficients. So it's going to do that real quick. And after it does that, it's going to run the simulation. But as you can see, calculating the coefficients can take a little bit of time. And it looks like an assertion failed. I'll go and figure out what this is, but something went wrong. Oh, I know what's going on. I told it to use a CUDA object, and this isn't a CUDA binary, so it failed. We don't have any singularity containers pre-built for Cardioid. I know how to build them though. If you want one, I can give you one. I will advise you though that Cardioid is simple enough that I would just compile it by hand instead of going through singularity containers, particularly because Cardioid binds MPI. And you guys may be way better than me, but I have never been able to get MPI to bind properly through a singularity container interface. And on top of that, you have to plug into CUDA and a very specific architecture dependent CUDA as well. Yeah, but I'm open to that if you want that. And I think that's it. I think we are about out of time. Yeah, the we can have a whole separate conversation about getting MPI to work through the container interface. If you really want to look into it, I would look at what the E4S people are doing as part of the XSL computing project. They have a nice interface to handle all those binding events across container boundaries by doing some run elf patching of the binaries. That would be the way that I would move forward. Also, if you were going that route, I would not use OpenMP, I would use MPeach, because what everyone is using for doing the bindings is binary compatibility that's built into different versions of MPeach. As far as I've seen, OpenMP has kind of fallen out of favor on that front.
Okay, we're getting close to the top of the hour.
Yeah.
Thanks, Rob. That was a really interesting and helpful walkthrough and demo.
Yeah.
I
don't believe we have a paper that covers all the optimal changes to the code. I can document it though for you. I have something I've written for the melody. I just got the pandemic happened, and then everything kind of got dropped on the floor.
Yes, as it does. So we've recorded this session. We'll put together a video of the recording through the SC21 website fairly soon. And I think, Rob, you're able to provide some slides as well for
yeah, I'll send you the slides in a couple of days. Yeah.
Thanks.
Yeah.
Thanks for
no
that.
problem.
And thanks again, Rob, and everybody for participating and for the great questions.
Yeah, thank you.